And then you get what they call a foam of carbohydrate, and that's what the popcorn is. And so, that's the mechanism that the water inside has to build up to a critical pressure, and then it explodes. Well, the chemists were doing X-ray crystallography and the whole line of things that chemists do, and what they found were the best poppers were the ones with the most ordered crystalline carbohydrate in the outer hull when it was heated, not the ordered hull when it was cold. And they couldn't tell this when the carbohydrate, when they just looked at the corn before it was heated. But when the corn hull, they couldn't tell by just measuring the hull or anything like that. And the reason I'm telling you this, we're going into this story about popcorn, is that the importance of the carbohydrate, but also the importance of the structure, that the crystalline structure made it stronger. If you think about carbon, and coal is carbon, and it's sort of an amorphous crystalline structure, diamond is carbon with a very ordered crystalline structure. So structure makes a difference. I mean, you wouldn't want to flaunt, I have a beautiful rock of coal on my finger here, because the crystalline structure makes a big difference to the kind of properties of the element. And so because it was crystalline, it was much stronger. And so the bottom line is, and the take-home lesson for this is, that structure is essential for function. And what I have here is the structure of glycogen, which is really branched, the structure of different kinds of starches, which are not very branched. And the reason I'm showing you these structures is not to edify you or turn you into chemists or to make you think I'm a genius because I can show structures, but rather because structures mean something. And that by showing the structures you can perhaps learn something about how things work. An engineer wants to look at the structure of a bridge. He can get an idea of how worthy it is as a bridge by looking at the structure. You can get an idea of how good a molecule works by looking at the structure. By looking at the structure of glycogen, you can see that it's really branched and that the glucose molecules, there's lots of sites that the glucose molecules could come off. And so when we need to break down glycogen, it can be broken off pretty easily. We can use it rather readily as a source of carbohydrate. In contrast, if you look at starch, some of it is just a single chain. You have to cut the glucose off one by one by one. It's a rather slow process. So structure makes a difference here. And when we start getting into lipids, structure will make a big difference. And so, this is something for you to remember, that we'll be showing you structures, but it's important to know that the structures mean something to the function. Next step in glucose metabolism. Once, the last lecture we got to the digestion, that glucose and the monosaccharides and the disaccharides got into the lumen, were broken down, got into the intestinal cells. That's where we stopped, right? Remember? Long time ago. So they were in the intestinal cells as of last Monday. And so when glucose gets out of the intestinal cells, it goes directly to the liver. The blood from the intestine, instead of following what you would think is the normal pathway, goes directly to the liver. what the normal pathway is, say your blood to your big toe goes from the heart down to your big toe, it turns into veins, goes back to your heart. The blood to your brain goes out of the heart to your brain, back to your heart. But the blood from your intestine, the blood goes to your intestine right here, and out of your intestine to the liver, and then back to the heart. So the intestine is different than all the other places. It goes, the blood goes to the intestine, then to the liver, and then back to the heart. So the liver screens is the first pass for everything that goes out of the intestine, goes through the liver, most everything. Because, as you'll learn next week, there's two paths out of the intestine. One path to the liver, and the other path is what happens to fats. The fats go into the lymph, and the lymph goes directly into the bloodstream near the heart. Next week, we will have a guest lecturer for 2 lectures. I won't be here. It's Dr. Ryan. He does research in lipids. He's extremely knowledgeable in this area, and so he'll be giving 2 lectures in the field of lipids. He doesn't know about the test. He doesn't know about enrollment, but he's very knowledgeable about lipids. Okay, so we're back to carbohydrates. So the carbohydrates are in the intestine. They go from the intestine through the portal vein to the liver. And then the liver metabolizes them, and that's the next slide. So blood from the intestine and glucose go directly to the liver. And then the liver glucose has a couple choices of what the glucose can do in the liver. And it can then be used for energy. And glucose is important to the body. It's the main energy source, but it's particularly important for the brain and red blood cells because that's the main energy source for the brain. Other organs use other things, but the brain really uses glucose in the nervous system. So that's really, really important. So glucose, that's number one. It can be stored in the liver as glycogen. Glucose can be released into the circulation for other organs to use, or it can be stored as fat. This is a very, very minor pathway. Glucose generally does not go to fat. Okay, and we're going to talk a little bit about how blood glucose, the level of circulating glucose is kept constant. That's coming up. And so the functions of glucose in the body. So we've got glucose, we've got it from the digestion through the portal vein to the liver. Now it's out of the liver in the circulation. And in the circulation, it has some choices. It can be stored as glycogen either in the liver or as the muscle. It can be used as energy in any of the cells. A lot of the cells, particularly brain. It can be used to make glucose. One function of glucose is that it's called to spare protein. If you don't have glucose or a carbohydrate source, you break down protein. So it's called protein sparing. So if you don't have glucose, you will break down dietary protein first. And if you don't have dietary protein, you'll start breaking down your muscle mass. So you absolutely need carbohydrate, particularly for your brain. And if you don't have dietary protein, and then finally, you need glucose to prevent ketosis. You need approximately 50 to 100 grams of carbohydrate a day to prevent ketosis. And ketosis, you'll find out next week, is the incomplete breakdown of fat. It's when your brain or your breath smells like acetone. When you end up with chemistry lab breath. It's a product of inefficient fat breakdown. You don't want to develop ketosis. There's a lot of problems with the physiology. When you start developing ketosis, you're weak, you smell bad. Yeah. The ketosis is the breakdown of fat to ketone bodies. If you have sufficient carbohydrate, fat will break down to energy. But if you don't have sufficient carbohydrate, fat will break down to ketone bodies. So that's the reason to have efficient fat breakdown so that you get energy from fat. You really need carbohydrate so that you can efficiently use your fat. Questions? Everyone okay with that? Yeah. Yes, that is one of the arguments against the Atkins diet. I mean, another argument against the Atkins diet is if you look through history and you think about the Atkins diet is carbohydrates are bad, insulin causes poisoning, all this fat storage, all this other stuff. But if you look through history, if you look through all the, needs to be dealt with, civilizations, the Chinese civilizations, the Babylonians, the Aztecs, the Incas, the Egyptian civilizations, all the civilizations, they have lived for thousands of years on carbohydrate-based diets. There's only one civilization that has survived on a meat-based diet, and that's the American Eskimos, the Native Americans, the Inuits. That's it. All the other civilizations have prospered on carbohydrate diets. Okay. For thousands of years and millions of people. Okay. Okay, hormones regulating glucose metabolism. Okay, there are lots of them, but we're only going to talk about two now. Insulin, that results in glucose going into cells. Insulin, glucose going into cells, right? It's synthesized in the pancreas, and it controls transport into cells. Do we have a question? I hear a question. For me or for someone else? Okay. Glucagon. It's the opposite hormone, going in the opposite direction. That also is synthesized in the pancreas. It elicits the release of glucose out of the glycogen stores. So insulin, after a meal will cause the glucose to go into cells, and when you're hungry and you need energy source, glucagon will cause the breakdown of glycogen. Okay, on tests, I always have these two words mixed up, glucagon and glycogen. Glucagon, glycogen. Okay? Don't get them mixed up. One's a hormone. Glucagon, glycogen is the carbohydrate source. I always put that on the test. I always tell people I put it on the test. Okay. Shall I tell you how many people always get it wrong? A glucose tolerance test is when a person is given a dose of oral glucose, say, I think it's 50 or 100 grams of glucose in a little drink, and then after a fast, and then their blood glucose is monitored at intervals. So their blood glucose is monitored before they take the drink of glucose, and then at intervals after they take the glucose. This is what's called a glucose tolerance test. And this is a normal one, that the normal level of glucose is somewhere in the range of, the normal fasting level is somewhere in the range of 70 milligrams per deciliter. You don't have to know that number. But we normally have, even on a fasting state, we normally have circulating glucose because we need it all the time. We'll get glucose from somewhere. Even if we've been fasting for 4 days, we will still have circulating glucose because we need it. But after a meal or a dose of glucose, we will have our blood glucose go up rather abruptly, and then insulin will come in and then cause the blood glucose level to go down. And then after a while, the blood glucose level will go down even a little bit below what was the normal fasting level, and then it'll go back up a little bit. So the profile of insulin will sort of look pretty much like this, but a little bit offset. The insulin will increase and then go down too. Some people call this hypoglycemia. It's not hypoglycemia is the condition that diabetics go into when glucose is really low. This is the condition when you're feeling a little munchy. This is not hypoglycemia, but your glucose is a little bit low. So this is the normal situation when after a meal or a dose of glucose, your blood glucose goes high and then insulin kicks in and causes it to go down. Okay? And so this is what happens. We have, our normal blood glucose in a range. So after a meal it goes up, and then we have elevated blood glucose, and then the pancreas releases the insulin. Insulin causes glucose to be transported into cells. The cells convert glucose into glycogen, and then the blood glucose goes back down to normal. Okay? And then the blood glucose goes down again. Once it gets a little bit low, we need more blood glucose, and so we have the opposite reaction. So blood glucose is low. Right here, goes down. So we have low blood glucose. The pancreas releases the glucagon, the opposite hormone, and then we have breakdown of glycogen to glucose. We have increased synthesis of glucose, and that causes glucose to go back up to normal. So we have the two hormones. Glucose goes up, we have insulin, causes it to come back down again. Glucose goes down. We have glucagon, causes it to come back up again. And the two are always acting one against the other to keep the glucose level normalized. Question? I'm sorry. If you're fasting for 5 days, you will still have glucose in your body. You will be breaking down fat, and you'll have ketones in your body, but you will also be breaking down your body protein, and you'll be using that to supply glucose for your brain. Yeah, so fasting is not good for your muscles because you start breaking down muscle to supply glucose. Okay, diabetes type 1 is when your cells of your pancreas no longer can make insulin. This is, maybe 5% of the population have this kind of diabetes. It's a destruction of the beta cells of the pancreas that they can no longer make insulin. It's It runs in families. They think it's kind of an autoimmune disorder. It's not strictly genetic, but it's got genetic features to it. And the consequence of it is you can't store glucose. And that you always, after a meal, you have too much. And then sometimes you have too little. And so the treatment is very careful diet, and people need insulin injections. With too much glucose, you have tissue damage. Too little, your brain's starved of energy. So type 1 diabetes is really very dangerous. This is the disease when people, go into diabetic coma. They need amputations. They have kidney failure. Type 1 diabetics have a lot of health problems. And so here's the same picture again, our normal glucose profile. But this is what an insulin-dependent diabetic profile would be. They would have a much higher fasting glucose level, and after a glucose load, their blood glucose would just soar sky high. And then it would stay up really for a much longer time because they would not have the insulin to make the glucose go into cells. So you see a very dramatic difference between a normal glucose tolerance test and an insulin-dependent diabetic or type 1 diabetic glucose tolerance test. Okay? So these type 1 diabetics don't make insulin. They need insulin injections. Type 2 diabetics, they do make insulin. This is most of the diabetics now, but they are what they call resistant to insulin. Their insulin receptors no longer respond to insulin the way they should. And it's a spectrum of disorders. Sometimes it can be mild, sometimes it can be very severe. So they have the insulin, and sometimes they're pumping out lots and lots and lots of insulin, but their insulin receptors are not responding. So the treatment for these people is, number one, exercise. Exercise makes the muscles respond to insulin much more actively. Yeah? Right. Some type 2 diabetics do take insulin because that's when the mild ones don't need it, but when they get really, really severe, the pancreas totally gives out.
So even though they're insulin resistant, they still need to take insulin for the little bit of time they have left for it?
Right. Because the pancreas just blows itself out. Yeah. Yeah. It is a wide spectrum of disorder. And so the treatment is you start early, you start with lifestyle treatment, you start with exercise, you start with diet, and then you have to go into drugs. Yeah, question. No, it's one or the other, but type 2 can progress from mild to very severe. Yeah. It's very prevalent in overweight people. Why? Because that's a hard one. There's a lot of fat cells, and they have so much stored energy that it's hard for the the cells to find actual places to store more energy. I mean, the insulin is trying to store more energy in the cells, and what it's seeing is the cells are all filled with energy. Yeah, with fat. Yeah. And so, it tries to go into glycogen, and glycogen is maxed out. And next, it's trying to store the extra energy into fat, and now the fat is getting kind of maxed out. Yeah, into fat. Right. Question? Yeah. Mm-hmm. Yeah. Yeah. First of all, a mild type 2, this was the question, can they just stop making insulin? What happens is they become insulin resistant, and so they start making some more insulin to try to compensate for being resistant. And then they make some more to try to compensate for being resistant. Then they make more, and then finally the pancreas just gives out. They can't make any more. And then it's a crisis, and they don't make insulin anymore. What happens to all the extra insulin in there? The insulin, there's high circulating insulin, but if the cells can't respond to it, it's just there in the bloodstream. There's no receptors for it to bind to. Too much sugar, there are a lot of disadvantages to having too much sugar in the bloodstream. I'll get it, okay? Here's the consequences of having too much sugar in the bloodstream, is you end up with damage to the large blood vessels because the sugar starts binding to things. It'll bind to protein, it'll bind to the blood vessels, it'll bind to the small blood vessels. In the eyes, it binds to the little blood vessels in the retina. It will also bind to parts of the cornea. It causes blindness. It binds to the filtration units in the kidney, and it ends up causing kidney failure. It causes it binds to parts of the nerves and causes them not to function appropriately. And so people with diabetes typically lose feeling in their extremities, and typically their toes. And if they the typical scenario is they lose feeling in their toes, and they get a small infection in their toes, but they can't feel it. And then they have a lot of circulating glucose in their blood. And so with a lot of glucose in their blood and a small infection, the small infection turns into a big infection, develops gangrene, and then diabetics are very susceptible, and then they have an amputation. First it's the toes, and then it's the foot, and then it's the knees. And so diabetics, you see a lot of diabetics sitting there with no legs. And then they develop kidney failure. And, uncontrolled diabetes is really bad. The way they test for diabetic control is for glycosylated hemoglobin. They'll look at a sample of blood and look to see how much of the hemoglobin has sugar bound to it. And this is a picture over time of how good the glucose control has been. You don't want your hemoglobin that's supposed to be carrying oxygen to be bound up with sugars. No, it's not working right. Okay, so hemoglobin or excess sugar causes a lot of problems. Susceptibility to infection, damaging a lot of tissues and organs, and the whole spectrum of disorders. So type 1 is the really bad one. Type 2 starts off rather mild but can get really bad if it's not kept in check. It's kept in check by diet at first, and then there are drugs to keep it in check. And finally, after it's gotten really bad, there's insulin and drugs for it.
So home blood sugar monitors are measuring milligrams per deciliter.
Home blood sugar monitors will measure glucose, but I'm not sure what units they use. More questions? Yeah. I don't, I don't know. I don't know. Yeah.
I know there's two types of diabetes. There's diabetes insipidus and diabetes. Do you know which one is which type?
Both of these are diabetes mellitus. Okay, accusations against sugar. We have to go quick. Sugar causes obesity. Okay, who's sticking that sugar in your mouth? There's no evidence that sugar increases appetite. They've done those kinds of studies that does, sugar make your appetite change as compared to other foods. It's no different than other foods for affecting appetite. And carbohydrates and protein have the same caloric value. Fats have a higher caloric value. But there is some evidence that diet drinks increase appetite, which is strange. But yes, there is evidence. Okay, sugar causes heart disease. That's not true. Not for most people. Some people seem to have a very strange response to carbohydrates. But the usual risk factors are inactivity, smoking, and genetics. So that's not sugar's thing. Sugar causes misbehavior in children or criminal behavior in adults. Long before you were born, there was this trial called in San Francisco, and there was this thing called the Twinkie defense, that some guy alleged he murdered someone else because he was eating Twinkies and it made him criminally insane and he couldn't control his behavior. He got off. But it's had a bad name ever since. But they've found in controlled trials with children, that's not substantiated. But that's been a big joke about the Twinkie defense. Wrong one. Sugar causes dental caries. Yes, it does. Brush your teeth. What happens is that the bacteria in your mouth digest the sugar. By now, after all my lectures, you should know that we are covered with bacteria. It's in our mouth, on our skin, in our digestive tract, in our stomach, in our intestines, everywhere. We're covered with bacteria. And the stuff in our mouth, as soon as we get sugar in our mouth, the bacteria are going to start working on it. Okay, and when they digest the sugar, it will turn to acids, and the acids start eating on our teeth, which aren't good for our teeth. But that's one problem. But the second problem, and this is for older people, is that they're found a relationship between gum disease, the periodontal disease, and heart problems because of the bacteria that cause the gum disease are infecting are related to the problems with heart disease. So, start now and don't get yourself involved in gum disease. It's a bacterial infection, and you don't want to get a systemic infection. So it is related. And then finally, sugar causes cravings and addictions. There is such a thing as a food addiction. It's real. I mean, there's an alcohol addiction. That's real. It's not necessarily a carbohydrate addiction. It's a food addiction. And it causes an increase in brain neurotransmitters such as serotonin. But what it ends up as, in the same way as an alcohol addiction, is it's a self-medicating behavior. That there's a chemical imbalance, that the serotonin is imbalanced. And you use something. You use alcohol, you use food, you use something to try to modulate that imbalance in the brain chemistry. And addictions are real. And it's the self-medication to try to deal with the addictions. So, alcoholism is real. And so the important thing is to realize that it is a brain chemistry deficit, and that it needs to be dealt with, as a medical problem. That's it. Thank you.